Our part 3 of the lecture on chapters 25 and 26 with this animation on classification schemes.
In the early days of biology, all organisms were classified as either plants or animals. Single-celled eukaryotic organisms such as algae and paramecium were assigned to one of the kingdoms according to whether they are photosynthetic or not. Fungi and prokaryotes were grouped with plants because fungi are sedentary and prokaryotes have cell walls. A five-kingdom scheme was devised because some organisms don't fit well into a two-kingdom system of classification. The simple cells of prokaryotes set them apart in their own kingdom, Monera. Organisms in the other four kingdoms all have eukaryotic cells. Fungi are not photosynthetic, so they are given their own kingdom. The kingdom Protista is an assortment of eukaryotes that don't fit into the other kingdoms, mostly unicellular organisms such as paramecium, unicellular algae, and multicellular algae. The five-kingdom scheme divides living things into two fundamentally different groups, prokaryotes and eukaryotes. New data suggests that two kinds of prokaryotes diverged early in the history of life, and thus there are actually three major categories of organisms. A new classification system assigns more significance to the ancient split by creating a level of classification larger than a kingdom, a new category called a domain. The two domains of prokaryotes are bacteria and archaea. The domain eukarya encompasses all of the kingdoms of eukaryotes. Taxonomists are in the process of sorting out various kingdoms of prokaryotes within the domains bacteria and archaea, and also splitting the protists into several kingdoms within the domain eukarya.
Okay, so now that we've seen the way that we can classify these, we'll see how we can depict them in a cladogram or phylogenetic tree. These are what make up the connection between classification and phylogeny, because it represents a hypothesis about their evolutionary relationships, and each branch point represents a divergence of two species. So a phylogenetic diagram, also known as a cladogram, is constructed from a series of dichotomies, and it reflects the hierarchical classification of taxonomic groups as well, as they are nested within more exclusive groups. So a phylogeny is determined by, again, the collection of evidence such as fossils, molecular data, morphologic features, and most systematic, oh boy, I can't say that word. The people who apply systematics use cladistic analysis, which was developed by a German entomologist by the name of Hennig. And they analyze the data in terms of the sequence of branching so that it will symbolize historical chronology. This picture shows how the branches are actually defined in some terminology that you should know that goes along with the branching patterns. So first of all, each branch point is where the lineage diverges. And so you can see here at 1 we have one divergence into these two groups. And here we have a divergence again that branches into more. And sister taxa are groups that are closely related and share an immediate common ancestor. So here's the immediate common ancestor. A rooted tree includes a branch that represents the last common ancestor of everybody in the tree. So in this case, if you have this ancestor identified, it's a rooted tree. A basal taxon diverges early in the history of the group and originates near the common ancestor of the group. So this is the common ancestor of the taxa A through G right here. So this branch point is where we have our first divergent. A polytomy is a branch from which more than two groups emerge. So in this case, we have a divergence here and a branching of more than just two groups. We have three here, and so that's called a polytomy. And again, here's our basal taxon, which is the group that diverged early in the history of this entire phylogenetic tree, and it originates near the common ancestor. This would also be called an outlier. Here's another tree here that shows the connection between classification and phylogeny. Here we see the sister taxa are right here between these organisms. These species here are sister taxa, and then these two are as well. This is a rooted tree because it shows the original common ancestor. The basal taxon in this case is the panther. It actually diverged early on. And there are no polytomies in this particular tree because we don't have more than three branching off at one point. Here are some terms that you should be familiar with. When we're talking about similarities of characteristics, what we're talking about are homologies if they are based in their shared ancestry. So that means for some particular characteristic to be a homology, it needed to come from the same common ancestor. So in the case of the forelimbs of mammals and the forelimbs of birds, those are homologies. They're homologous structures because they came from a similar shared ancestor way back in evolutionary history. The contrast is an analogy, and in the case of an analogy, the similarity in structure or morphology is due to convergent evolution, such as the wings of birds and the wings of insects, or in the case of these two moles, the Australian mole and the North American mole, they both are animals that burrow, and so they, in their own separate evolution on different continents, they evolved, and natural selection favored an adaptation that allowed them to be good at burrowing. So they share characteristics that are similar, but they are analogous characteristics because they experienced convergent evolution. Convergent evolution occurs when there are similar environmental pressures in distinct species, and natural selection produces similar adaptations in those organisms that come from different evolutionary lineage. So here are some more comments to help you understand the difference between homologous and analogous characteristics. So bat and bird wings are homologous as forelimbs, but analogous as functional wings. Their analogous structures or molecular sequences that evolved independently are also called homoplasies. And a homology can be distinguished from analogy by comparing fossil evidence and the degree of complexity. The more elements that are similar